So we have seen what is a cell cycle in our previous video. Okay, so what is interface? Okay, so what is G1, what is S, what is G2, how a cell gets prepared to enter into mitosis. Okay, so mitosis is like the most important phase for a cell to get divided into two daughter cells. Okay, so every cell, okay, except the your sex chromosomes like X and Y, okay, except these two, all other cells will undergo mitosis for their replication or like to multiply. Okay, so this mitosis is like the most important phase for all the cells during its replication. Okay, so now we will see what happens in this mitosis. Okay, so mitosis actually starts with the end of your S2 phase. Okay, so once the cell is get ready, okay, so it has copied all its organelles, it has copied its DNA, and it has accumulated enough energy and nutrient sources for the mitosis, and it makes sure it is ready. Okay, once it's ready, it will give a signal, then your mitosis phase starts. Okay, under your mitosis you have like 4 subphases. Okay, so it's called as prophase, metaphase, anaphase, and telophase. Okay, sometimes you have like metaphase, you can classify as pro-metaphase. Okay, but commonly we have prophase, metaphase, anaphase, and telophase. Okay, so these are all the 4 phases. Okay, so what actually happens in these 4 phases? Okay, so that's what we are going to see now. Okay, so I'll show you like one more diagram, okay, for your better understanding. Okay, so at the end of interface, that is G2, you know that your cell is ready, okay, for division. Okay, but it has a nucleus, okay, inside which you have a copy of 2 copies of your DNA, okay, which got from your S phase. Okay, but now you have to divide this all your organelles which have made its copy as well as your DNA, so that you get 2 daughter cells. Okay, so for this there is one important organelle is going to do some function. Okay, so it's called as centrosomes. Okay, so centrosome usually will be present in your animal cells rather than your plant cells. Okay, plants will have an unique component to do the same job of your centrosomes. Okay, so the centrosomes will have 2 centrioles. Okay, so you could see here there are 2 small objects. This is called as centrioles. Okay, so the centriole is the key factor in this mitosis process. Okay, so the first subphase in your mitosis is prophase. Okay, so what actually happens in your prophase? Okay, so there are 4 important things. Okay, so try to remember, because if you know the prophase, you can easily understand the anaphase. Okay, because whatever happens in prophase, the exact reverse will happen in your anaphase, sorry, in your telophase. Okay, so try to remember this prophase so that you can easily understand what happens in telophase. Okay, so first prophase. Okay, so in prophase, initially inside your nucleus, your chromosome is going to be there, but initially the chromosome will not be visible to eye. You could see here, okay, difference between this image and this image. Okay, here it's like in a decondensed form. Okay, so decondensed means it will be like in a thread-like structure. Okay, like a very large coil of thread-like structures. Okay, so it's like in a decondensed form. Okay, so the first thing is this chromosome gets condensed, okay, to acquire a proper shape. Okay, so to have a proper structure, okay, in a so that it visible to the eye. Okay, so in that state it gets condensed. Okay, second one is your nuclear envelope starts disappears. Okay, so surrounding your nucleus you have a thing called as nuclear envelope. Okay, so this nuclear envelope slowly started to disappears. Okay, so nuclear envelope disappears and your chromosomes gets condensed. Okay, then the third change happen in your nucleus is the nucleolus gets disappeared. Okay, so 3 changes happens with respect to your nucleus. First is your nuclear envelope disappears, then your nucleolus, okay, that also disappears. Third, your chromosome is going to get condensed. Okay, so 3 changes happen in your nucleus area. Then in the fourth one, what happens? You have a centrosomes. I told here. Okay, so the centrosomes will have 2 centrioles. Okay, so here one centriole will move to one direction and another centriole will move to the exact opposite direction. Okay, So it's like locating in the two poles. Okay, one if it's in north pole, another is going to be in the south pole. Okay, so likewise it starts to move. Okay, and there is going to be a formation of spindle fiber. Okay, so here you could see there is like a thread like structure formed between these two centrioles. Okay, so it's called as spindle fibers. Okay, so the spindle fiber is like a connecting thing between these two centrioles. Okay, so this arises from this centrioles, which is from a centrosomes. Okay, so this happens in prophase. So now what happens in metaphase? Okay, so in metaphase, before going to the metaphase, just we'll see this word as pro-metaphase. Okay, so you could see this moving centrioles reaches opposite poles. Okay, then the spindle fibers have been like completely formed, okay, to all the areas. Okay, and you could see here the nuclear membrane has been completely disappeared, nucleolus also has been completely disappeared, on all the fragments of your chromosome, okay, has been like arranged in the center part of your cell. Okay, so now what happens in metaphase? Okay, so in metaphase what happens is your chromosomes arrange in the metaphase plate. Okay, so this center line, okay, which we call it as metaphase plate. Okay, so in this line all your chromosomes will align, and so in the center part of your chromosome you will have a thing called as kinetochore. Okay, so this particular kinetochore is going to get joined with the spindle fibers. Okay, so this kinetochore is going to get joined with your spindle fibers. So once this kinetochore part, okay, that is the center part of your chromosome, gets joined with the spindle fibers, all these will align in this center metaphase plate. Okay, you could see this complete image. Okay, so both the centrioles are in the opposite direction, the spindle fibers are completely formed, and all the chromosomes are arranged in the metaphase plate, and all these spindle fibers are connected with the kinetochores, that is the center part of your chromosomes, okay, in the particular structure. This is metaphase. The third subphase is anaphase. Okay, so what happens in anaphase? Okay, so here in anaphase your chromosomes are going to be splitted into two equal halves. Okay, so usually the condensed form of your chromosome, okay, this is like decondensed form, this is your condensed form. Okay, so a condensed form of your chromosome will have daughter chromatids. Okay, so two pairs of daughter chromatids. Okay, so which will have actually four arms, 1 2 3 4. Okay, so this is a single daughter chromatid, this is a single daughter chromatid. Okay, so likewise you will have a pair of daughter chromatids in each of your chromosomes. Okay, condensed chromosome. So during your anaphase what happens, one pair of daughter chromosome chromatid and another pair of daughter chromatid will be pulled away from each other by the spindle fiber. Okay, so spindle fiber starts gets pulled by this centrioles towards itself. Okay, so this centriole will pull this spindle fiber towards itself, and this centriole will pull this spindle fiber towards itself. So because of this pulling action, what happens, this the center part of the kinetochore attached to the spindle fibers pulls and rips away these two daughter chromosomes away from each other. Okay, so similarly same pattern happen in all the chromosomes. Okay, so on both sides a single pair of chromatids are going to be separated. Okay, so this happens in your anaphase. Okay, so the last stage of your mitosis. Okay, so what happens here? Okay, it is called as telophase, which completes with the cytokinesis process. Okay, so what is actually telophase? Okay, so during telophase, so now what happens, whatever we mentioned in the prophase, the reverse of it is going to occur in the telophase. Okay, that is the centrioles initially which formed will move to its original position, and the centrioles will slowly start disappearing. Okay, then your nuclear membrane starts to reappear, your chromosome gets decondensed. Okay, initially here and all it was in condensed form, so now it's again going to become into this form, okay, which is a decondensed form. Then nucleolus also will reappear. Here it disappeared. Okay, so here nucleolus disappeared, here reappear, here nuclear membrane disappeared, here it will reappear. Okay, then here nucleolus disappeared, here it will reappear. Okay, so all the exact opposite will happen in telophase. Okay, and so already you have different organelles separated. Now your DNA is also separated. So on both sides, or the single thing will be saved. So here one mitochondria, Golgi apparatus, one DNA. So likewise, each set will be given on both the sides. Okay, so this is your telophase, but still your cell is not completely divided. Okay, so for that it has to undergo the final stage called as cytokinesis. Okay, so what is cytokinesis? Okay, so now everything is separated, but still the cell has to divide it into two daughter cells. Okay, so this happens in the process cytokinesis. Okay, so this cytokinesis already we have referred in plant cell versus animal cell topic. Okay, so the cytokinesis happens in two different ways in plant cell and animal cells. In the case of animal cell, a cleavage furrow. So this is we called as cleavage furrow. Okay, so it's less constructs in the center line. Okay, then this divided into two daughter cells. Okay, this will happen in the case of animal cells, but if you take in the case of plant cell, this cleavage furrow will not form. Okay, instead a cell plate will be formed. Okay, a central cell plate will be formed, which will finally divide this into two daughter cells. Okay, so from interphase it enters into prophase, then metaphase where all align in a central line, then in anaphase it splits your chromosome, and in telophase all the copied things will be segregated into two sites. Okay, finally in cytokinesis it forms two daughter cells. Okay, then again these two daughter cells is going to undergo the same cycle followed by the parent cell. Okay, so this is how a cell cycle works up. Okay,